All right, so for this series of lectures, I want to talk about the subject of genetics and behavior. And obviously, when we talk about behavior, we're really talking about neural systems, right? We're talking about the genetics of neural systems and things that happen as production of proteins and use of proteins in neural systems. So I thought I would first just go over a little bit about neural morphology, brain morphology, just to kind of bring those things to the forefront so we know what we're talking about and reading about. Then we'll talk about some genetic disorders that relate to behavioral disorders. And there's many, many types. We're going to cover maybe some schizophrenia, which we've already talked about, briefly. Some forms of dementia. There's multiple forms of dementia. We'll talk about some of the relationships of genetics to that, as well as other things, and we will, I think what I'll probably do is break these down into 4 lectures so they're not so long each. And then I want to close out with a couple of slides in the last lecture about animal models, how we actually go in and utilize lab systems to understand even human behaviors and try to break them down and focus them. But for now, what I want to talk about is neural morphology. Just to remind you a little bit about some of the structures there that you've probably seen in comparative anatomy or other places. But here's a human brain. Okay, they weigh somewhere between 1.3 and 1.4 kilograms on an average brain. Lots of convolutions, etc. And they, of course, changes over time as we age from young to old, we gain or lose these convolutions, as well as when we have certain medications, certain other things that can happen. A huge number of neural cells, somewhere in the order of 100 billion cells called neurons that make up a brain. And there are even more support cells that are not neurites, not transmitting signal, but have protecting that signal either through sheathing or other things. These are called support cells, neuroglia cells, and there's trillions of those because you need multiple ones for each neural cell. Each neural cell connects somewhere between 1,000 to 10,000 other cells in various ways, branching patterns and sorts of things, and these are called synapses. Okay, so we have a number of synapses, and these will actually change over time. And we're going to talk about pruning at one point. And pruning means how many of these synapses that you actually have or break down. The brain does have 2 hemispheres, the right and the left hemisphere, and lots of activities are located, portioned out into at least the human brain, from the cerebellum in one area, maybe up to the cerebrum in others, or in various places. One thing that doesn't have good support, even though we have right and left, and we clearly things like the optic nerves cross, and so you see on the right what you're really seeing on the left eye, things like that. The sort of widespread concept that they're right and left-brained people doesn't have a lot of support by the data. Right and left, somebody might be, say, if you're right-brained, you're more analytical. If you're left-brained, you're more artistic, or you're more musical, or you're more something else. There's no really good support for that. The reason being is that, if you can see in this, kind of can't really read this chart, but areas that all are spread all over the brain, all over the hemispheres that do different things. So you really can't be isolated. If you have issues on one side of the brain, there's still other things that go on. So there's really not a lot of support for that concept that's out there. Now, each neural cell, each neuron, has a number of components. Obviously, as with many things we've talked about, I don't expect you to memorize these components. I just wanted to give you an image of what's going on. You have, of course, a typical cell. Cells, these have DNA in them, right? Neural cells, for the most part, do not replicate once they're mature. That's one of the reasons that you have spinal injuries and other things are things that happen in the brain where you get very little ability to replace them. Now, that doesn't mean that they won't sometimes reconnect in other ways, right? Because if you have thousands of connections, you may have ways of going around, and people can relearn things when there's brain issues. But neural cells, only a very, very few types of neural cells will actually go through a mitotic process and reform again after everything goes on. Interesting things like songbirds almost have an entire brain in the spring when they go in mating phase. Particularly the males, and the females, but the brains of males will almost completely redo themselves. They'll just replicate and reform and reshape, and then over summer and over the later periods, those shapes will change. Human brains don't. Okay, very few places do we have any types of replacement. There are some, but not many. But they're typical cells. They have, they have Golgi bodies, they have ribosomes, they have lots of mitochondria, ER, and all the other things that you would expect to see. But what they also have are these extensions that go out, these dendritic extensions that come in, that connect to other cells, and these synaptic ones that go out. All right. They're sheathed. Again, these are cells. These are those support cells, those neural glial cells that support the outside of various functions. And then, of course, you have synaptic regions where they come in very, very close contact with other neurons, and they have spaces where there's firing. That's how the connections go on, is through these firing phases. And we'll see later that this is one of the areas that really causes lots of issues with things like dementia and things, is that we block these connections, either through putting things out there or by just having the actual pores and things that are necessary for the neurotransmitters to move don't function properly, or the neurotransmitters don't get produced properly. But sometimes it's just merely a fact that these become gummed up and you really can't transmit. Anyway, these are neural cells. Length of neural cells, a few centimeters, maybe a few millimeters in some cases. I think the longest one's maybe a meter that comes out of the medulla moving down. That's actually out of the brain. The longest nerve in the body, a human body, is the sciatic nerve, which runs from the lower back down to the toes. That's the longest one in a human. I think what might be the longest neuron out there, maybe the giant squids. They certainly have long ones. Giraffe necks, people usually, a lot of times will say giraffe necks because you've got connections there. You have to run all the way to the back of the body. Single nerves. Probably, I would say something equivalent to the sciatic nerve that's found in, say, something like a blue whale might be the longest. That's one of the biggest mammals, and by far, and they have a sciatic nerve, just like you do. So I suspect that's probably one of the longest that are out there. But we're talking about brains, human brains, and so we're talking about things that are in millimeters or smaller units. Okay. Now, at these junctions, these synapses that we call them, basically what you're going to have are neurotransmitters being pushed across. Okay, and there's typical processes that you would see in cells where these neurotransmitters are produced. They're usually, they're proteins, so they're produced somewhere through the process of transcription, translation. There are, as we may remember, there were kinesin and other things that run along, and those have these long channels and microtubules regions where kinesin will bring proteins away out this way. And of course, in reverse, you have dynein that will transport it back. So you have these systems that move these neurotransmitters once they're produced, and this usually in the center part of the cell. They'll be moved out as needed. These neurotransmitters then have a reaction once there's a stimulation of the neural site, and they'll fire across, and they're small enough, they're tiny to be picked up and actually cause a cellular response, either by physically being moved into the other cell through a receptor or simply stimulating a receptor. And again, receptors are just more proteins, right? They're just other parts of the cell. There are multiple things that can happen to them. They can be returned and reused. There's this little pattern of a transporter port right here that will grab them and pull them back. They can be broken down and used within the cell itself, or even externally. There are degrading enzymes that are found in these interstitial areas that will break them down, and then they may be potentially reabsorbed in the system or not. But that's basically how the synaptic system works. Now, what are some of these neurotransmitters? Well, there's a large number of them. They tend to be fairly small chemical structures. I mean, these are ring structures. They're not particularly small, but they're tiny in size. You really think about them. And some are very, very small. And some are things that you're probably familiar with, like glycine right here. Amino acid, it signals. So glycine forms. There's other, adenosine and things that break down and transfer in. Tryptophan, it's not on here. Tryptophan. Tryptophan is a precursor to serotonin. Some of the ones you probably are very familiar with from biology or from your own life are things like serotonin. Okay, serotonin is found is what many antidepressant therapeutics are designed to mimic that, at least to be taken up by those receptors. And so either when you have that, you have very many cases you have a serotonin decrease naturally if you increase it or stimulate it with pharmaceuticals. That's what some of these antidepressants are. Most narcotics, as it says, affect release and reuptake. Then the other side of that is dopamine. Dopamine is highly involved in things like the reward pathway and also voluntary action. This is the one that things like cocaine and habituates and things. So we'll talk about some of that later, and we talked about it earlier, but we'll come back to it again. Is that we got these pathways, and the people that are highly susceptible to these chemicals get a high reward, and then eventually that reward breaks down. They need more and more and more, and eventually, and sometimes it can even reach to a level, particularly with habituates, where it becomes a toxic system to the body. Norepinephrine, they're produced in various parts of the brain, and some are external to the brain. Some are actually internally produced. Interesting that serotonin functions in the gut, the central nervous system, and other places as well. Acetylcholine is a very important one, and probably the most common, most well-studied unit that we can see, and it has all sorts of imbalances. The point of this is that when we think about these little neurotransmitters, depending on which part of the neural system you're in, what part of the brain, or in fact, what part of the body that you're in, and different ones play different roles. And they may, overlapping roles in various places. You may have serotonin and things like dopamine in the same areas working at the same time in different ways, counteracting each other or enhancing each other. Okay? Just those examples of those. Dopamine pathways, that's the ones that are in blue here. And dopamine pathways are found commonly in the core area of the brain. There's a big concentration of them found there that fire out of the hippocampus, obviously, are in there. And then the frontal cortex is highly impacted by it. But at the same time, serotonin, which is a more broadly applied one of these neurotransmitters, is found throughout the brain, but it also overlaps at the same time in many of these regions. Again, dopamine, it's reward, motivation, pleasure, euphoria, things like that. Fine-tuning motor functions, so where you can grip fine or fast, is controlled by this, and it controls things like compulsive behavior. Again, it's one of the ones that's very important as a pharmaceutical. Serotonin, mood, depression, mood. Serotonin is you don't get enough serotonin, you don't sleep well, that sort of thing. Memory processing, sleep, and cognition are part of those. So those are all neurotransmission systems. Now, as you can imagine, since these are proteins and they're produced by a genetic pathway, if you have failure of something, you have a mutation, chromosomal aberration, something wrong with the pathway, regulatory change, then you can have a change that affects these and therefore will affect behavior, right? Which is what the topic of this talk is about. Right? So just some examples of where we know that there's some relationship. And again, these are not the only places. But acetylcholine, which has to do with memory and learning, is involved in things like Alzheimer's disease. Okay, and what it does is the lack of it causes some deterioration of these synapses and these grading of it. Okay, dopamine is interesting in that oversupply is involved in some forms of schizophrenia. Now, we're going to talk about schizophrenia, and you'll see that it's a massively complex structure, but oversupply is linked to that. So having too much dopamine, undersupply linked to things like tremors and the mobility problems, the lockup that you see in many people with Parkinson's disease. Serotonin, an undersupply is linked to depression, and that's why many antidepressants are designed to mimic either physically, go in and activate serotonin production or mimic serotonin, so they do it. And others, norepinephrine undersupply can be found in people that have, mood swings, depressive mood swings, and things like that. Okay, so these do have effects, and obviously things, glutamate. It's a neurotransmitter involved, and that what's interesting is that oversupply can overstimulate the brain, leading to headaches, things. And that's many of us MSG. People avoid MSG because I've got a headache. Well, actually, it's you're getting basically a glutamate response from overstimulation in the brain of activity that we interpret as a headache. But for people who have serious other underlying conditions like prone to migraines or prone to seizures, it can actually drive it into that. Okay. Now, how do we know all this stuff? And we're going to show you some huge diagrams about all the pathways and things that these neurotransmitters can affect, or genes that are involved can affect by making of proteins. Well, that's done in a number of ways, of course. First off is there's, whole genomic analysis. Nowadays, certainly more commonly, we will look at expression. So we'll build expression libraries, RNA libraries, and various tissue types and see what's going on, or we'll stimulate an area of the brain and come back and say, okay, whoa, in those cells in this period of time with stimulation, what was being produced, what was not being produced as far as RNA, and therefore the proteins. Once we can do all that, and various ways we do it, we then have a pathway analysis where you come back and you think about all the different proteins and therefore all the genes that are involved in production of those proteins and where they map out to. And then ultimately, particularly for pharmaceutical things, you come down to a candidate gene approach. And this is just one where, where you identified in the dopamine pathway all of these different components that play a role. And here's here COMT gene. COMT gene is found on chromosome 22. In the long arm here, in this fairly active gene region, big complex gene region here, where actually there's genes on both sides, both strands, one of those places where it's very common to have overlapping introns on one, be an exon on the other, and you read both sides. But in this region of Q11, 21 to 22 here is where this particular gene is found on this tiny little chromosome that's over here. And so once we understand that, then we'll go back and we'll pick another one on your found the X chromosome or whatever, and start building these big connections. And we're going to see those big connections, and you've played with some of them because you've been asked to go out and look them up on the web. Okay. All right, well, that, just kind of an overview of neural structure, some of the neurotransmitters. And so now what we want to talk about in the next series of lectures are some of the outcomes of this. What's with genetics? What happens if we do something with genetics? What do we end up with? And we'll just use 4 big examples, very common. So we looked at and saying there are lots of different disease, outcomes, but we'll pick some that are very, very some complex and some just kind of sort of interesting. That'll be in the next lecture.